Hello everyone, thank you for the introduction. I'm Anastasia Tsagkarakou, and today we will discuss some results of one of my projects about the structure-based drug design for galectin-3. Galectins are a family of carbohydrate-binding proteins and have a specific affinity for beta-galactosides due to the structural and sequence similarity in their carbohydrate recognition domain. The family of galectins is divided into three subclasses according to their CRD distribution. The first one, the prototype galectins, contains only one CRD. The second one, the tandem repeat galectins, contains two CRDs, one in the terminal and the other one in the C-terminal domain, connected via linker. And the third one is the only type, galectin-3, the chimera one. Galectin-3 is the only member of the galectin family with an extended non-lectin flexible N-terminal region connected to a C-terminal CRD that we can see in the picture. Galectin-3 consists of approximately 135 amino acid residues that fold into two antiparallel beta sheets of 6 and 5 strands, jointly forming a beta sheet sandwich structure. S1 to S6 strands make up a concave with specific ligands can bound. This concave, this binding groove, can be subdivided into subsites A to E. The most conserved subsite is the subsite C, is the highly conserved among all galectins family, harbors the beta-galactoside, whereas the less conserved residue accommodates the sugar residue next to beta-galactose. In contrast, subsite A, B, and D are more variable and thus specific for each galectin. Galectin-3 is one of the most well-studied proteins of the galectin family. Numerous previous studies have indicated that galectin-3 may be used as diagnostic or prognostic biomarker for many types of diseases and conditions. Participates in a myriad of diseases, particularly those associated with chronic inflammations and fibrosis, and thus it has become a validated pharmaceutical target. Several galectin-3 inhibitors, small molecules, saccharide molecules, or bigger natural products, has already been synthesized from public or private companies for therapeutic applications. The thiodesaccharide compound that we can see is a potent thiodegalactoside for treating idiopathic pulmonary fibrosis, but terminated in the phase 2B of the clinical trials. But there are some promised results in the phase 2 clinical trial for COVID-19 pneumonitis. The other compound, the GB1211, is currently in phase 2 clinical trials as a potential therapy for liver cirrhosis and cancer. So in general, the main experimental procedure that we follow for the structure-based drug design, not only for galectin-3, but in general for the protein that we want to use. Firstly, we discuss with our collaborators, organic chemists. They synthesize and design really potent ligands. Then we are trying with a lot of structural biology approaches, mostly X-ray crystallography and NMR, ligand-based and protein-based NMR, to understand how the ligand binds in the protein that we study. Next, we are trying to find the affinity, the KD or the KI if it's an enzyme. For galectin-3, we use the ITC, isothermal titration calorimetry, to calculate the thermodynamic parameters of the complexes. Then, having all these results, we're trying to understand how this ligand binds, how is the affinity, and then we discuss with our collaborators. We have a lot of fights because we always ask different adding groups that it's really difficult without understanding the difficulties of the organic chemistry field. And then again, we start again the whole circle in order to find ligands with a high affinity and selectivity for the protein. Following exactly the same experimental procedure, today we will discuss and I will show you the results of those 4 compounds. As we can see, the 4 compounds share exactly the same core of 1 to 4 thiodesaccharide, but we have differences in the adding groups. The first one is a bis-aryl-triazole compound. The second one contains one added group of 3-fluorophenyl-triazole and one group of carboxyacetamide moiety. The other one contains only one fluoronin-triazole, not binding in the beta-galactose part, but to the ring next to beta-galactose. And the last one, as we can see, it doesn't share exactly the same core because it's not an 1-to-4 thiodesaccharide, but an O-lactosyl reference that we use in order to understand the differences. with a hydroxy benzoyl group. Here you can see the thermodynamic parameters of the complexes. Firstly, we can see that the reference all-lactylcyl compound has a low affinity at 12.9 micromolar with a huge entropic penalty. In parallel, we can see that the compound JZ814 and JZ744 are more or less equipotent with an affinity at 53 and 78 nanomolar respectively. But we can see that the bis-aryl triazole is more entropically favorable than the 6-carboxyacetamide moiety. At the same time, the bis-aryl triazole favors binding and leads to higher potency than the other compound with only one 3-fluorophenyl triazole moiety. The 4-fluorophenyl triazole moieties and the fluorine mods are preferable in general than the hydroxybenzoyl moiety. But in general, we can see that the binding is enthalpy driven, and if we can see this compound is the candidate of the clinical trials. They don't share exactly the same core because this one is 1-4 thiodesaccharide and this one is 1-1 thiodesaccharide, but they have exactly the same adding groups and they are mostly equipotent, but we have differences in the entropy. For the structural characterization of the complexes, firstly we employed NMR experiments. Ligand-based NMR approaches were first employed to investigate the molecular details of the interaction, allowing to map the linkage epitopes and therefore to identify the region that comprised the binding interface with the carbohydrate recognition domain of galectin-3. Saturation transfer difference, NMR confirmed the interaction and enabled identification of the ligand epitopes recognized upon binding to galectin-3. Comparison of the ASTD NMR spectra with the reference spectrum revealed distinct signal enhancements indicating selective ligand recognition. The highest enhancement of STD comes from the beta-galactose, and then as we can see we have different percentage of STD enhancement of the aromatic groups of the adding groups. Following exactly the same ligand-based NMR approaches, here we can see that the analysis demonstrates preferential binding of galectin-3 to the different compounds that we used. And in order to look further, what's going on, we performed crystallography, X-ray crystallography. The data collection was carried out at the P13 outstation in Hamburg. All ligands were found bound on the carbohydrate binding side of galectin-3. The beta-galactose binds exactly with the same manner in all the 4 compounds, but we can see a lot of differences in the adding groups. As we can see, and we said earlier, the beta-galactose to all 4 compounds for exactly the same direct hydrogen bond with subsite C, and the side chain atoms of the residues of the well-conserved subsite of galectin family. If we can see clearly in the table, we can see differences in different interactions with the water molecules. One important thing, if we want to design selective and high affinity ligands for galectins, and more specifically for galectin-3, is that in the carbohydrate binding group we have a lot of conserved water molecules. So the thing that we have to do is to remove those water molecules and design some compounds and the adding groups to perform direct hydrogen bonds with the side chain atoms of the residues of galectin-3. And here we can see that the first compound forms only 4 direct hydrogen bond interactions. This is really important because, as we can imagine, and if we remember the first table with the thermodynamic parameters, we saw that this compound has a preferable entropy than the other compounds. Moreover, we found a lot of differences to the adding groups. As we can see here, and here the fluorophenyl moiety of the first compound J814 and J7474 extends deeper into the pocket formed by arginine 144, which is non-conserved residue in the galectin family, and alanine 146 in the subsite B. And it can form a lot of fluoronic bonds with the side chain atoms of non-conserved residues. On the other hand, the carboxy moiety of these compounds forms polar contacts with non-conserved residues of the subsite E of galectin 3, and moreover p-stacking and arginine P interactions. The compound JZ848 can form only p-stacking interactions with the arginine 1HC of subsite E of galectin 3. So, having all this information, we can understand that the fluorine bonds and the contacts in the subsite E between the ligands and the galectin 3 are really important for the high potency. As regards the last compound, the reference one, the O-lactosyl compound, we can see here that we don't have a really good electron density map in the adding group. All these compounds were tested to galectin. Oh, yes, sure. Yes. We did a comparative structural analysis of this compound to galectin 1, 3, and 7, and at least for this compound, we saw some differences when this compound binds to galectin 7, forming direct hydrogen bond interactions with the subsite E of galectin 7. So maybe this compound can be a lead compound for galectin 7 and not galectin 3. So in conclusions, the present study offers a new starting point for the development of potent and selective inhibitors for galectin 3. As regards the 1 to 4 thiodisaccharides, so significant affinity enhancement over other 1 to 1 TDG based galectin 3 ligands or 1 to 2 thiodisaccharides. These findings establish two design principles. The first one is that the engagement of the subsite B and D by well-positioned aromatic regions on some other adding groups is essential for nanomolar potency. And second, that the entropic cost of binding must be minimized through rigid pre-organized substitutes capable of displacing the ordered water molecules of the carbohydrate binding group. The future of optimization might be just to think maybe the C2 or C3 substitute will possibly enhance the binding interaction with subsite E of galectin 3. I would like to thank my supervisor Salva Silippo and Professor Tony Molinaro from our research group, our collaborators of course from Prague, and all of you for your kind attention.